So thank you very much for this opportunity to be here. Development and clinical severity of irritable bowel syndrome are linked to psychological stress, mucosal microinflammation, and a significant substance P nerve positivity. Peripheral corticotropin-releasing factor and neuropeptides, such as substance P, are involved in stress-induced gut dysfunction. Moreover, intestinal eosinophils play a role in intestinal inflammation and have been identified as a local source of CRF in ulcerative colitis. However, the contribution of eosinophils to IBS pathophysiology remains unknown. For this, our aim is to identify the role of mucosal eosinophils in diarrhea-prone IBS and to evaluate the mechanism underlying the stress response using an eosinophil in vitro model. To study intestinal mucosal eosinophils, we recruited patients with IBS with diarrhea phenotype according to ROM3 criteria and healthy volunteers as a control group. In all participants were excluded food allergy, gastrointestinal infections, and inflammatory disease. We assessed the clinical data, including the abdominal pain and the IBS syndrome score, the stool consistency, the number of bowel movements, and the psychological stress and depression levels, all with validated questionnaires. A jejunal biopsy was obtained and proceeds for the analysis of eosinophil activation by gene expression for genes involved in eosinophil chemotaxis, anti-activation, and secretory activity by real-time PCR, and for the analysis of the ultrastructure by transmission electron microscopy. CRF location and content were evaluated by immunolabeling in transmission electron microscopy. To evaluate eosinophil response to neuropeptide, we employed the eosinophilic cell line 15HL60. This cell line, after differentiation with the sodium butyrate, was stimulated with substance P, or with carbachol, that is an analog of acetylcholine, or with LPS as a control of non-neuropeptide, and proceeds for the analysis of eosinophil activation by gene expression for eosinophil-specific genes, and for the evaluation of secretory activity by immunofluorescence. CRF location and content were evaluated by immunofluorescence and FACS. As a result, in human study, mucosal activity evaluated by gene expression showed a decrease in eotaxin 2 and in eotaxin 3 gene expression in IBSD group, suggesting less recruitment of eosinophils into the jejunal mucosa. We also observed an upregulation of SNAP23, that is a protein related to vesicular transport, in the IBSD group, suggesting an increase in secretory activity in this group, despite no differences in BAM2 and syntactin4 were seen. The analysis of the eosinophilic cationic proteins showed no changes in MVP gene expression between groups, but showed a decrease in ECP, in EDN, and in EPO gene expression in IBSD group, which suggests a less inflammatory state of eosinophil in this group. However, secretory activity was more prevalent in IBSD group, as indicated by piecemeal degranulation with selective fragmentation of cytoplasmic granules and higher frequency of vesicular structures, indicative of activation. By contrast, the control group showed a well-preserved electrodense granules and few secretory structures. The analysis and quantification of the eosinophil activation was evaluated in IMIG by the analysis of the granule number and the area of the eosinophils, the number of lipid bodies, and the degranulated area. There were no differences in the number of lipid bodies between groups and in the number of granules per eosinophils, but there were an increase in the granule area and in the degranulated area from eosinophils in IBSD group. Among mucosal immune cells such as plasma cells, macrophages, dendritic cells, lymphocytes, and mast cells, CRF was identified only in eosinophil granules. CRF was identified by gold electrodense particles in both the healthy and the IBSD group, but the analysis of the total protein content of the CRF revealed a higher amount of CRF per granules in eosinophils from IBSD patients. Moreover, the amount of CRF per granule positively correlated with the granulated area of eosinophils. The amount of CRF also positively correlated with the clinical symptoms, with the intensity of pain in IBSD and the severity score, and with the depression and stress levels, and also with the number of bowel movements and the stool consistency. So to gain insight into the mechanism underlying the stress response, we evaluated a neuropeptide stimulation in the eosinophilic cell line. For this, we first evaluated the substance P, the acetylcholine and LPS receptor in this cell line, as shown in these representative images. We then evaluated a time course gene expression response after stimulation with substance P, carbachol, and LPS of the eosinophilic cationic proteins. And as we can see in the graph, there are neither changes in MVP gene expression during time nor in the other eosinophilic cationic proteins. However, we evaluated secretory activity at protein level, and we observed a relocation of SNAP23 and BAM2, two proteins related to vesicular transport. After stimulation with substance P and carbachol, we see a relocation from the cytoplasm to the plasma membrane, indicative of stimulation and degranulation. We then localized cytoplasmic CRF expression in this cell line, and we also observed, after stimulation, a relocation from the cytoplasm to the plasma membrane after stimulation with substance P and carbachol, but not after stimulation with LPS, indicative of secretion. Moreover, the analysis with FACS revealed a decrease in the total protein content after stimulation with substance P and carbachol during time, but not after stimulation of LPS, indicating CRF release. For this, we can conclude that CRF is stored in the jejunal mucosal eosinophils, and its increase correlates with clinical symptoms and psychological factors in IBSD. That despite the low pro-inflammatory state of mucosal eosinophils, its secretory profile and increased CRF granular content suggest a role of eosinophils in IBSD pathophysiology. And similarly to mucosal eosinophils, in vitro response to substance P and carbachol, but not LPS, suggests eosinophil contribution to IBSD through neuroimmune mechanisms. I would like to thank you to my group for their support, especially to Javier Santos and Maria Vicario, and thank you for your attention.